Okay, now we're going to move on to case study 3, which is the intensive care unit gut microbiome. Now, why should we be interested in such a thing? Well, the intensive care unit is home to the sickest patients in the hospital. There's a wide range of patient needs there. Almost all of them need ventilation. They're having intravenous fluids. Often they're getting so-called parenteral nutrition, so they're getting fed through an intravenous tube. They have a high use of antibiotics. There's all sorts of other things going on. They're often getting proton pump inhibitors to prevent them aspirating acid from their stomachs. All sorts of care options going on in terms of them being preventing bed sores, looking after their hygiene, all that kind of stuff. And the point is that these are highly prone to infection, largely because they're very sick, but also they're laying flat on their back. Typically, the normal physiology of clearing respiratory secretions, normal gut motility, and particularly all those needles going through their skin providing breaches of the normal barriers to infection. All that means they're really sitting ducks for infection. And what we knew before we did our study was that there was a crucial role for the gut microbiota in this, in that those organisms living in the gut, it acts there as a reservoir of pathogens and of antimicrobial resistance. And there were some hints that having low microbial diversity might be linked to adverse clinical outcomes in this setting. And it's generally a rule of thumb when you look at gut microbiomes that the richer and more diverse the gut microbiome, the healthier it is considered to be. And loss of gut microbial diversity is generally considered to be a bad thing across the board, not just in this setting. And the way we're thinking, this is perhaps the epicenter of resistance and infection, this gut microbiome. So typical healthy microbiome patient out there in the community wandering around doing their own thing. That becomes a kind of sick microbiome once you start applying antimicrobial resistance pressure, selective pressure. And so you end up selecting for resistant things anyway. Whatever's there that's not resistant will get destroyed. But the other problem is that once patients are in the hospital, they're in touch with this hospital microbiome. They can get colonized with multi-drug resistant hospital clones. Maybe that those are very much minority components to begin with, but once you start applying antibiotic selective pressure, they can proliferate in the gut, and they can then spread from the gut to other compartments, so into the bloodstream, into the patient's lungs. They can contaminate the cannulas and so forth. And because there's this large biomass of AMR, multi-drug resistant super clones, if you like, superbug clones, those can spread around within the hospital environment, within the intensive care unit, and you can get cross-infection or at the very least cross-colonization of the same clones across. And so this scenario was one that there was a lot of evidence for it already, not down at the level of genome sequencing, but there was a lot of evidence to suggest this kind of stuff was going on from conventional microbiology. And what we thought was, what we could do is we could restore the healthy gut microbiome, perhaps via probiotics or fecal microbiota transplants, prevent infection, prevent the spread. And the idea of fecal microbiota transplants is one of these things that's quite fashionable in many areas now as a way of restoring a healthy microbiome, short-circuiting maybe what would be a slow process of restoring normal health, and as it is used for infection with Clostridioides difficile, but maybe it has a wider role, and that's what we were thinking here. And so our research questions are, what's happening to this gut microbiota in these patients? How does it respond to antibiotics? Do the antibiotics really select for pathogens? And what we were interested to do was also look at a methodological issue. Can we, instead of having to grow everything from the gut and do lots of onerous conventional microbiology, couldn't we just sequence the gut microbiome directly by sequencing DNA from the feces? And as I say, the question was, is there a case for an intervention here, like fecal microbiota transplants in this setting? And I should say this was a multi-institutional collaboration where there was the hospital in Birmingham, Queen Elizabeth Hospital. There was an NIHR-funded Surgical Reconstruction and Microbiology Research Centre. I was working at the University of Birmingham when this setup started out. We made use of the CLIME infrastructure, and we finished this off at the Quadram Institute with staff there. So in fact, just to show you again who was involved, a clinical microbiologist colleague, Beryl Oppenheim, worked at the hospital. ICU physician was Tony Whitehouse, and myself, my postdoc Anu Ravi, did the ecology and genomics with a bit of help from Willem van Scheek, who works at in the University of Birmingham. So you can see here that on the left there's the Queen Elizabeth Hospital building, and then our shiny new building, the Quadram Institute. We had 275 samples from 30 patients, and we had the relevant metadata collected as well. And I should point out that collecting that metadata was quite an undertaking, and shouldn't be dismissed with a wave of the hand. That was a real job of work for those that did it. We did shotgun metagenome sequencing on the NextSeq, and then we looked at this in terms of what was there in terms of taxonomic classification. We looked to see what we could conclude about antibiotic resistance, and we could also, in some cases, even get down to the level of strain level resolution. What we found was really quite surprising. I mean, quite shocking that actually 2/3 of the patients experienced low gut microbial diversity. I was expecting to see an effect, but this effect was so marked. And in fact, 1/3 of the patients had really very, very low, ultra low microbial diversity, where in fact what we saw in 75% of patients were episodes of gut domination by MDR pathogenic strains. So strains that we knew caused problems in this kind of setting: multi-drug resistant Enterobacter, multi-drug resistant Enterococcus, multi-drug resistant E. coli, multi-drug resistant Pseudomonas, Candida albicans, fungal pathogens. We saw these emerging in the gut as major dominant forces in a very clear way. And as I'll point out in a moment, we actually saw transmission of epidemic clones between patients. So in many patients we saw a picture like this, where when we looked at the DNA sequences and we classified them taxonomically, we found that a huge component of the gut microbiota was taken up by a single pathogen. In this case, Candida albicans, fungal pathogen, which is inherently resistant to antimicrobial agents and had taken over. And we know in intensive care unit patients, Candida albicans take over the gut, but then it can start to spread more widely into the bloodstream, covers the skin, gets into lines and so forth. So, and this patient was a lung transplant patient, so a very immunosuppressed and highly susceptible patient, and given a whole range of different antibiotics, so tazocin, cotrimoxazole, voriconazole. So the voriconazole was supposed to dampen down fungal pathogens, but didn't seem to have been much effect on Candida in the gut here. One of the things that is worth stressing is that we do see a kind of consistent approach, though. So when we take multiple samples from the same patient, we do see the same organisms coming up and roughly the similar kind of picture. And so it changes from sample to sample, but those changes tend to be cumulative. And that was satisfying. It wasn't totally chaotic. We saw consistent patterns in this, which was gave credence to the kind of methodologies we were using and the decisions we could make on them, conclusions we could draw. Sometimes patients did get away with it, and we didn't see weird, horrible, pathological things happening. So here's a polytrauma patient where basically had a fairly healthy gut microbial diversity retained throughout their stay in ICU. So even though they'd had initially erythromycin and tazobactam, they maintained this diversity. Although one slightly odd thing was that we did get Methanobrevibacter, which is actually not a bacterium, even though the name suggests it might be. It's an archaean, which is a different form of life entirely from bacteria. And again, like fungi, these are resistant to antimicrobials. So that was kind of blooming in the background. It's not thought of as a pathogen, but you do wonder, is it entirely benign as it blooms there? And the other thing you can see is that Bifidobacterium, which is considered one of the good bacteria, that did disappear quite quickly in this patient. The other thing to say is a trauma patient is one where we would perhaps expect to see the least disruption. So a patient who's had a lung transplant or a patient who's had liver failure for a long time, they're going to have been around healthcare premises for a long time. They'll have multiple courses of antibiotics. So their gut microbiota, even before they came in the intensive care unit, may have been damaged. Whereas a trauma patient, if someone's in a road traffic accident, they'll have a good normal gut microbiota, and then they go into hospital with that. And so that might explain what we're seeing here. We did sometimes see unexpected things. So typically when we got a multi-drug resistant pathogen dominating the gut, it stayed there for a period of time, long periods of time. But here's an example where we had Enterococcus faecium in this patient, and it really was, in some cases accounting for 100% of the reads. It really had the gut microbiota, the feces was a pure culture of this organism. But then it looked like there was a change, and we had Streptococcus thermophilus, which is a common component of probiotics that are given. So we assume that someone administered probiotics to this patient, and then we did get a restoration of normal gut diversity over time. So it's not always doom and gloom forever. Sometimes you do get recovery. And one of the things that we really don't know about is how recovery occurs after the patients leave the intensive care unit. How often do they restore normal gut diversity? When we looked at strain level diversity, particularly we looked at E. coli, we could see to begin with in this patient who had a bleed that they had two strains, one that was sensitive and one multi-drug resistant. They got given these broad spectrum antibiotics, and what we saw was that the multi-drug resistant strain hung around and then bloomed towards the end of the study period, whereas the sensitive strain actually disappeared during the middle of things, and then it did come back towards the end. But it's interesting that we see the strains come and go. The other thing that we saw also was vancomycin resistance in this patient came and went as time went on. And that blue there is Enterococcus faecium, which is where that VAN-A gene, which causes vancomycin resistance, was residing. Oh, and there's basically the drug-resistant strain coming back. And this just shows you a range of patient profiles, and you can see a very similar sort of picture that generally, once they get one of these large populations of a multi-drug resistant organism established in their bowel, it hangs around for long periods of time. Here's a list of all of the different pathogens that we saw, the kind of clinical conditions, and sometimes we got clinical samples with the same pathogen. But one thing that we did notice that we focused in on was there were a series of 3 patients here, 51, 54 and 55, that all had Enterococcus faecium growing in abundance in their gut microbiota, and they had different kind of histories. One of them had ST elevation and infarction, one of them had alcoholic liver disease, one had a subarachnoid hemorrhage. But in each case, they had a bloom of Enterococcus faecium. And when we looked at that particular strain in detail, we found there were very, very few SNPs separating them. So what we were seeing, therefore, was movement of this, and when we looked at where they'd been on the ward, the A's are actually the beds and the P's are the patients. We can see that they were in adjacent rooms in a very much geographically confined space here, and although we didn't see this particular strain transferred from those patients out to other compartments and actually causing clinical disease, we clearly are getting nosocomial transmission of that strain between patients, which is a bad thing, something we don't want to see. Now, what we wanted to do was try and establish, is there any kind of clinical parameter that is linked to this loss of diversity? Could we find evidence that, for example, that diarrhea or use of particular antibiotic agents or something called the SOFA score, which is a general feature of how sick they are in general, did any of these actually link to the gut microbial diversity? What we've found was rather disappointing. It's a very messy picture because it's a very heterogeneous population. But one thing we did find was the use of the broad spectrum agent meropenem was linked. And so this is one of the bad guys here. You give them meropenem, you tend to see a significant effect on gut microbiota. And that was the main statistically significant finding there. What we did find, though, was that some of the beneficial commensals, I already mentioned that one about bifidobacterium, they tend to disappear in most patients. Faecalibacter is another one. Is this something that we can dismiss and say, well, they're not necessary? If we look at studies on patients elsewhere, we often do have these organisms earmarked as good things, but they disappear. And as I mentioned, commensals, even things like Methanobrevibacter smithii, bagged as a commensal, but it does tend to dominate the gut in some cases. So what does that mean? And this chart here just shows you the domination of the gut by different organisms in different patients and different samples. And you can see by the kind of black smears in the middle there, Enterococcus faecium dominated in multiple patients. If you look up the top there, E. coli also tended to dominate in some patients. And right down at the bottom there, Methanobrevibacter smithii, and particularly Methanobrevibacter as a genus, did actually tend to dominate in a lot of patients. Whether that means anything in terms of, is it contributing to their continuing illness, we don't know, but it's certainly it raises that interesting hypothesis to be tested. So in conclusion, what we found when we did this was shotgun metagenomics provided detailed information on the microbiota trajectories and pathogens seen in ICU patients. We did see a loss of diversity. We saw a loss of commensals. We did see clear effects of meropenem. We saw clonal blooms of these MDR pathogens, and we saw cryptic transmission. If you're interested to read more about this, the paper's published. It's out there. There's the link to it, and you can follow up everything there. One of the disappointments was that we did not establish a clear case for fecal microbiota transplants or even for probiotics. There's a hint that maybe probiotics might work once you've stopped the antibiotics. Obviously, probiotics are going to be susceptible to antibiotics, so giving them while you're still giving the antibiotics is probably not going to be helpful. One interesting sideline is perhaps phage therapy. So given that we know that there are sort of dominant clones, it may be that these do provide almost a fixed target that you could design a phage cocktail. And this is something that is coming to the fore now. And Willem van Schaik in Birmingham, I think, is following this up now with a PhD student, trying to see what they can do in terms of looking at what the bacteriophage profiles are in these patients as well, and what interventions might be possible. And clinical metagenomics is showing promise for the future here. It actually did deliver useful information. Now, a skeptic might say, well, you could have just grown those organisms, they will grow readily and got the same results. But yeah, we've just cut out some of the steps along the way, and we've got a much rounded picture of what's going on in the gut microbiome by going in for shotgun metagenomics directly. And the smart money is on doing this with something like the MinIron, where you also get long read lengths as well, and it's a easier target. So what I'm going to ask you to do now is, again, final act of student participation. So if you follow that link there, that'll take you to a trace from one of our sequencing runs. It's one of the reads in there, and you can look at that in what they call ChronoView, which is an interactive pie chart where you can click on it and you can focus in on different components of the microbiome. If you use the back arrow, you can go backwards and look more generally, or you can go down and click on things. So take a look at that one and see, what do you think is going on here? What can you see from analysing that metagenome just using this very simple chronoplot that might strike you as unusual or notable from that particular one? There's another trace you can look at as well and do the same thing. And in fact, if you want to, you can just go and look at any of the traces within our particular bio project and explore any of those results in an easy and interactive fashion. So I'll leave it to you to just go and have a look at that and get a feel for yourself as to what you can learn from these kind of analyses. That's me finished now. If you have any questions and you want to ask me them, please email me and I'll be delighted to get back to you. My email address is mark.pallen@quadrum.ac.uk.